So we're just getting to our speaker for tonight. I'm very lucky to have wonderful coworkers. So I have to admit my base job is at Fisheries and Oceans Canada, and I've asked Michelle to join me tonight. So Michelle Paleczny is part of the Coastal Marine Ecosystem and Research Program at Fisheries and Oceans Canada. She'll probably be also hearing about her coworker Sarah as well. They both really enjoyed field work, exploring the BC coast, sharing their discoveries, and you're going to hear a lot about, well, protecting the nearshore and intertidal ecosystems coastwide. So, Michelle, when you're ready, take it away.
Okay.
Oh,
thanks.
And I'll just say for everybody, if you have questions, I'm sure Michelle would be happy to answer them at the end, or if they're really pressing during the talk, like maybe raise a hand, or you can put them in the chat, and I'm happy to reach out anything that people aren't comfortable saying themselves. Sound good?
Absolutely.
Thank you.
Yeah, thanks, Heidi. Thanks for inviting me to speak, and thanks for tuning in, everybody, and sharing your evening. I'm giving this talk on behalf of myself and my supervisor, Sarah Dudas, who isn't here, but she sends her regards. And sorry, I've got a funny setup here. Okay, here we go. Yeah, so first I just wanted to start out by acknowledging that I'm giving the talk from the traditional territory of the Malahat Nation, and all of our work happens on a whole bunch of different nations' territories, so throughout the coast. So just wanted to acknowledge that. And then my story in a nutshell is that I'm a biologist. I share an office with Heidi, and I've worked on ecological monitoring research projects with UVic, UBC, Parks Canada, and then more recently for DFO. And tonight, I'd like to use my time with you to tell you about what our little known section does to support marine conservation targets on the coast. And our section is called the Coastal Marine Ecosystems Research Team, CMARE, and we're within the Marine Spatial Ecology Analysis section that Heidi mentioned earlier, that both Heidi and I work in. So there we go. So Canada's marine conservation target, you've probably heard of this.
Sorry, Michelle, not seeing your screen yet.
Oh, really? Oh,
yeah.
I didn't hit the share.
That's okay.
Sorry about that.
I just know you got good pictures, so looking forward to them.
Yeah, thank you. I have like kind of a wonky setup, so I'm so glad you told me that. Okay, because I wouldn't have noticed. Okay, there we go. Now you see it, right?
No, I'm sorry. Oh, here it's coming. Yay. I'm seeing it. Yeah.
Okay, thank you. So there's my first slide, and that's got like our program name on it, which is CMARE for short. And then this is Canada's marine conservation target that I just mentioned, to protect 30% of Canada's oceans by 2030 within these different protective measures. So there's marine protected areas, national marine conservation areas, national marine wildlife areas, and marine refuges. So there we go. Okay, so our work, mine and Heidi's work, a lot of it is related to supporting these marine conservation targets, supporting achievement of them. And then in my smaller CMARE program with Sarah, our role is to provide information related to conservation needs. So for example, on the distribution and abundance of taxa, like on their ecological significance, on emerging threats such as invasive species. We also, develop and evaluate monitoring methods and technologies to support marine conservation. And throughout this, we are also trying to contribute to reconciliation. And what that means has been evolving as we do our work, but so far it has meant applying OCAP principles of data sovereignty, asking for permission to visit sites, working with Indigenous partners and collaborators as equal partners, and supporting their research needs. So this is a list of key research projects that we're currently working on. And in the rest of this presentation, I'm just going to tell you about each of these projects and why we research it and how we research it. And this presentation is going to be more focused on why we're doing what we're doing and how we're doing it than on the results, partly because it's a newish program and partly because I'm newish to the program. So here's the list. I'll touch on these 4 different topics. And the first topic is coastal biodiversity. Research. Here we go. So marine conservation needs to know the what, where, and how of protecting marine ecosystems. So what we're trying to do is support that by developing methods and contributing to long-term monitoring in collaboration with First Nations. And I'll show you on the coming slides what I mean by coastal biodiversity research. This photo that you can see is a photo taken from a drone of an intertidal survey site, and you can see these blue lines delineate intertidal elevation zones, and then red dots are quadrats, which are random stratifiedly placed, I guess, throughout the sampling area within those tidal strata. And then so we use this drone image. We can show our survey site, but then we can also get information from this image and from the digital surface model about the site, and we can use the drone imagery as a monitoring tool itself. So when we're at the intertidal survey sites, within each of these quadrats that we've placed, we survey a whole bunch of different things. We survey the cover of seaweed, the substrate, the epifauna, so the animals living on the surface, and then the infauna, so the clams and worms and other invertebrates living inside the sediment. And we're developing a protocol in collaboration with Bridget Clarkson and other researchers on what to do, how to record seaweeds that are not possible to record to species in the field. So how to lump them into functional groups that tell us information about the ecosystem. And then at the intertidal sites, we also use cameras to do research on predators in the intertidal ecosystems like bears or birds. And this is done in collaboration with my supervisor, Sarah's grad student, Zoe Penno, as well as other members of the MC team, like Michelle Big here, and she uses wildlife cameras to monitor oil spills as well on the coast. She's developing a protocol for that. And then another thing we do at intertidal sites, we just recently started doing because we learned about this existing methods for collecting samples to learn about if forage fish are spawning at sites. So taking a step back, sand lance and surf smelt actually lay their eggs in amongst the sand and the pea gravel on some beaches at certain times of year. And not a lot is known about the timing and the abundance of the spawn outside of the Strait of Georgia area. So we had an opportunity, because we're visiting these remote locations, to collect some sand samples and then send them to a lab for eDNA analysis, where we can find out the relative abundance of DNA of those taxa, to learn about if they're there and in what abundance their DNA is showing up in the sediment. And that's a collaboration with Jacqueline Uard at Project Watershed. And then when we're at the intertidal sites, we also we use a seine net to collect fish and then learn about the abundance and diversity of fish that are there. And this was the first time I ever caught a snailfish, was this year, and I just thought it was so cute.
What?
And then at the intertidal sites, we also this is a new project, a new intertidal project that we started, which is long-term monitoring of rocky intertidal sites in Barkley Sound. And we're using the marine protocol, which it's a long-running protocol that started in the States. And we've adapted it to our sites, and we're monitoring anemones, barnacles, mussels, and sea stars in the intertidal at these established, like, long-term monitoring sites that we just established last year. And we're looking to see how these communities are changing through time, and we're going to use we are using drones as a survey tool there as well, and we're going to look at how the different survey tools, so like on the ground surveys versus drone surveys of some of the larger taxa like sea stars, compare in what we're seeing and how we can evaluate trends with the two different data sets. And these are also sites where there's a long-term diving. They're long-term benthic habitat monitoring sites that have long-term diving data. So we can compare subtidal data as well with intertidal data. Yeah. So then another project that the CMAR team is working on is Olympia oysters. So Olympia oysters are a species at risk, and they occur throughout BC, like southern BC, but their distribution is much smaller than it was historically, and the populations that are left are very patchy, and they're cryptic, and sometimes they occur mixed in with Pacific oysters, and they're also impacted by disturbance. So when you go to a site to monitor it, because they're so small and they often occur underneath other like rocks and oysters, there can be a big impact to boots on the ground surveying. So the objective of this research is to develop methods of estimating the population sizes at Olympia oyster sites, and comparing the boots on the ground survey to other techniques like drone surveys and eDNA. And the eDNA part is done by Sarah's grad student, Neha Asharia Patel. Sarah also started monitoring microplastics in seawater, because we know that microplastics are starting to show up in marine ecosystems. And we had this opportunity with this big ship that we use to access sites throughout the coast. It has a lab in it, and she set up this instrument that she developed in collaboration with Ocean Diagnostics in Victoria to filter the surface seawater that comes in from the front of the ship and into the lab, where it goes through this filtration instrument, which has a flow meter on it, so you can count how much water is filtered. And then using this tool, we've been able to collect water while the ship is underway over large parts of the coast. And then using this other tool that Ocean Diagnostics developed called the Ascension, we can also sample water at different depths from the surface. So this thing is deployed from the smaller dive skiff and then dropped down to specific heights or depths from the sea surface and deployed for set amounts of time or set volumes to filter the seawater. And it's filtered through these stainless steel sieves. And then we take them out. So the sieves are in these blue case housings. And then we take them out inside of this laminar flow hood. So we're using we've been developing these protocols because this is a new technique. So they figured out how to use this laminar flow hood to reduce contamination, environmental contamination of the samples. And then these samples are on the stainless steel filters. They're sent to ODI lab, and then ODI lab uses a type of spectroscopy to they look at the light reflected off of the plastic to figure out the type of plastic, and then they can also look at the shape of the plastic, and they report back, so the type, the shape, and then the quantity of plastic particles that are in the samples. And we've been working through issues with clogging of the filters because as you can imagine, in the spring when there's algae blooms, it gets tricky to filter out the surface seawater, filter out large enough volumes to get above the limit of detection without clogging the filters with plankton. So this map here is showing all of the red lines are places where we've done underway sampling on the coast. And then the blue dots are places where we use that Ascension sampling tool. And then the green dots on the north and central coast and west coast of the island mostly are places where we've done marine debris surveys. So that's where we walk. We use a protocol that was developed partly by the MC team in collaboration with a protocol that's used in the States, where we walk the tide line and we record the abundance and type of plastic. And then we pick up, and that's along a 90-meter transect. And then we pick up everything that we can fit into our boat before we leave and that we can detach, because sometimes plastic is very entangled. And we take it back with us. And whatever we can't take, we can also mark the location of on apps that exist, so that contractors who collect plastic debris can return to those sites or prioritize sites to visit to remove plastic in the future. So the last project that I wanted to tell you about that we're working on is it's on glass sponges, corals, and rockfishes, and this is my personal favorite. And we know that the reason I love them, and what we know, is that these complex bottom habitats are important habitats for a lot of other species. They support biodiversity. And one of the species or groups of species that they support are rockfish. So fisheries managers are interested in them for the conservation benefits to biodiversity as well as to rockfish. But bottom activities threaten these habitats because mechanical damage, so things hitting them can break them. They're fragile and they take a long time to grow back. And then also when sediment is disturbed on the seafloor, it can be resuspended into the water column and affect the ability of these filter feeders to pump water and to feed. So spatial management needs to know about the distribution of these taxa, these cold water corals and sponges, so that we can protect them. And the tricky thing is that they occur in deep water, so from about 20 meters deep down to hundreds, for some of them, I guess, thousands of meters deep. So because they're so deep, they're very difficult to map, and they're generally very data deficient. So we received a request for science advice through the Canadian Science Advice Secretariat, which is a peer review process to provide information on the distribution and abundance of corals and sponges in and around rockfish conservation areas in Jervis Inlet. So fisheries managers wanted to know where these corals and sponges were around rockfish conservation areas in Jervis Inlet, because they are looking at potentially improving rockfish conservation areas or other protection measures to include more of these types of habitats, which is part of marine conservation targets, or could potentially be. So how we did this with a big team of people was we used the ship, and then this is an ROV, and this is part of a bunch of work that is related to using ROVs to non-invasively monitor benthic habitats, which Heidi hopefully has shared lots about with you guys, because she is really the pro at it. So these are some examples of images that we captured, and you can see how this is a rockfish and this is a rockfish in just hiding in glass sponge. And I would like to show you just a couple of minutes of a video. Do you see the video?
I think you did this on purpose, but we did lose your screen. Okay, and it's
Okay.
back. Now
Okay.
it's back.
I think, okay.
Let's see if it played. Oh,
You see
there it's going.
See it playing? Okay. Okay, I think this will be people's favorite part, because it's my favorite part. But I just love getting to see this. So I hope it's playing well. But I'm showing the video in the Beagle program, which is what we use to annotate the video. So we make a mark where we see an individual rockfish, and we make marks where areas are covered like this in dense glass sponge. And you can see here below, these are examples of all the different things that we've marked. And this is my very favorite little patch of video. So right here, there's a little rockfish. And then there's a juvenile yellow eye up here that just disappears into the sponge. Sorry, I'm just figuring out something here.
I was just gonna say that was so great to see the footage. I think you were happy to be done there.
I just have a few more slides, but
Oh no, I meant with the video. Yeah, yeah. We're getting while you're sending that out in the comments. Oh, I mean, look at that art, beautiful. But yeah, someone is saying
oh
great
yeah.
videos. It's so wonderful to see these habitats. Like you can't describe it in words until you capture in video. So it's so great.
Yeah.
And one of the things they're asking is why are the rockfish and sponge reef like both on their own and together like so important for marine habitats?
Hmm. Yeah, sorry, I didn't explain that very well. Um, so they there's a bunch of different reasons. So they provide hiding place, like complex habitat that juveniles can hide in when they're potential prey for larger fish. Um, they provide food subsidy. So like especially they've found researchers have found when sponge occurs on rock substrate, there's this really neat mix of um, the sponge hiding places and then the food on the rock substrate that generalist predators like rockfish can eat. So they are kind of like a hub of of biodiversity in otherwise pretty bare seafloors. And then yeah, that would be in the case for both corals and sponges. Heidi, would you like, is there anything else as a bit more of like a sponge and coral expert than me that you would add to that?
Oh yeah.
Like, if
Yes.
so, just chime on in.
No, yeah, I think you did a great job. Like corals and sponges are just amazing for providing habitat, which provides everything from nursery grounds to more food to increasing the biodiversity of an area. But one of the things that why they're so important for kind of our groups, like Michelle's and mine's, is that these animals are old, like slow growing old animals that are like very fragile. So what you're seeing here is an animal that's actually built its body up out of glass. And then when it dies, because it's built its like this really cool structure out of glass, it can stay there and then more of the animals can settle on top of them. So they really build up really old complex structures where these glass sponge reefs in Canada have actually been dated almost 9,000 years old if you consider like from the base to the outside healthy sponge. But like I've just said multiple times, they make their bodies out of glass. So if it were contacted, you destroy this habitat, you decrease the biodiversity, you do all those sorts of things that then have negative effects that cascade all the way up to us and our food and our services that we get from corals and sponges. And then I think it's also similar, kind of the same idea with rockfish, like rockfish conservation areas, is that rockfish are a lot older and slow growing than a lot of like, if you think about salmon, we've all heard, salmon are on a 4-year time scale. That yellow eye is probably at least 100 years old. Like it's just the differences there. I don't know if that helped at all, but yeah, I think you
Yeah.
kind of nailed it. And just kind of seeing the beauty of these structures down there, like when they're growing up on top of each other. And if you were to, get a microscope out and look in between all those folds of glass, you'd see, crabs and snails and all those other things too living in them, which is pretty cool. Sorry, but we'll let you keep going on talk, and then there's even
No,
more questions coming in, which is great. So thank you.
cool. No, thank you for adding that. 'Cause I love listening to you talk too. You're so good at explaining it. It's awesome. Thank you. Cool. Do you want to do, I can show my last few slides and then we can go to the questions, or which one
Sure,
did you want
let's
to
do that.
first?
Yeah.
Yeah, okay. So I think I mentioned this already, but yeah, like this research that we are a part of is also a part of this like broader research that MC is doing on how to adequately monitor these benthic, these like, as Heidi mentioned, really important and diverse benthic communities. And so this team has developed a protocol for consistently collecting data from ROV footage, like when we're using ROV tools to non-invasively monitor these ecosystems, so without damaging them. And so one of the things we do is when we're watching the video, we record the amount of cover of live reef building sponge in 10-second bins of survey video. And I've included the scale that we use here to give you an idea. So when we're watching a 10-second bin, we see live reef building sponge, we assign one of these cover classes to it, and then we can map this data, so we can look at show maps of where these really high densities of reef building glass sponge occur on the seafloor. And then, in addition to monitoring the live reef building glass sponge, recording it, we're also making records of where we're seeing this damaged reef building glass sponge. So right here you can see there's some glass sponge that's knocked over, and then the general area is like broken and dead sponge. And it's difficult for sponge. So there's ongoing research on the conservation value of these areas where damage has occurred, because once the substrate is loose like this, it's difficult for young sponge to settle, because they need a hard substrate, as well as a substrate that doesn't have silt on it. So you can imagine in these places, it's difficult to find a hard substrate that's stable and isn't going to get covered in silt. So we observed this broken sponge rubble, we call it. And then we also observed corals that were broken and still, I guess I would call this like dying, like they're partly alive and partly dead. So this is also evidence of probably fairly recent mechanical damage as well. And then we can use that information to make maps that look like this. This is just one example. So we can show where areas of, for example, the high density live reef building sponge occurred, or high density paragorgia, those bubblegum corals. And there will be other maps and results that will be part of this, and it's coming out in a CSAS document in the spring. So that's everything from me. Thanks, everybody.
Ooh, finishing with a heart. Strong move. Love it. Yeah, thank you so much, Michelle. That's so great. I like knowing all the different kind of tools that our team is using. Like even Michelle and I work together, and I love just seeing all these different ways that we're gathering science. So I hear a bunch of people, or I see a bunch of people putting questions in the chat. I'm happy to read them out, but if you'd rather say them out loud. There's like at the bottom, there's a react button. You can like raise your hand, or you can just take yourself off mute and turn on your camera. You're welcome to do that. And so while people are getting brave, okay, Shiny, why don't you go ahead and ask your first question?
I was just wondering, like who or which organization do you contribute your data to, and like what do they do with it? Like how do they help conserve the marine wildlife?
Oh, yeah. Thank you for asking. I think I maybe a bit intentionally like didn't say that. I think so I work for DFO, but I always think of DFO, or I did before I worked for DFO, as like a fisheries, more of a fisheries focused organization. But we so we work within this marine spatial ecology section of DFO. And our data goes, a lot of the data is collected in collaboration with First Nations. So anything that's collected with them goes to them, or anything that they ask for. And then most of our data ends up on open data as well. So on publicly accessible eventually. And then a lot of it gets published in either primary like journal articles, or in CSAS, Canadian Science Advice Secretariat documents. Does that answer?
Yeah, just wondering like are there like people taking action? For example, like the rockfish, they're like have people been taking action with like against the rockfish? Like have they have they done anything?
Yeah, so that is the reason why I love that project, is because it's a very like management linked project. So in that case, the fisheries management team asked for this information so that they could include more of the sponges and corals within protected areas, which they're working right now on proposing, and then going through the engagement and consultation process with the public on. So
Yeah.
it does go a lot of our work is like, it's kind of a mix, right, Heidi? Like there's like a lot of management related projects that directly flow into management decisions. And then there's also, running alongside that is all this like research that goes into kind of anticipating what kind of information we're going to need, and how we're going to be able to collect it, to improve the effectiveness ultimately of marine conservation areas and marine conservation. Did you have something to add, Heidi, or?
Yeah, the only thing that I could say is that our team that Michelle and I feed data into, we're kind of like the science branch of Fisheries and Oceans. And then there's the management branch of Fisheries and Oceans. And before I started working here, I didn't really realize what a divide there was. So they are often asking us for science advice about it. And then they take our science advice and then they do the processes. For example, to establish a marine protected area, we give the science advice about where to protect, what's important, that sort of thing. And then they take advice with that, combine it with fishing data, combine it with Canadian public perception and input. And then they're the ones that design the marine protected areas. So we're one of the things that kind of go into it, the science biology advice. And then, yeah, it definitely does inform. So for example, in my program, we have like established a huge marine protected area offshore that includes, seamounts and hydrothermal vents. And we provided a science advice. They then took it and did all of that. So yeah, it's an interesting combination of just getting science out into the world, but also bringing it up through Fisheries and Oceans and other, like, for example, like to the First Nations, or First Nations led, Indigenous led marine protected areas or conservation efforts as well. So that it does feed into different things as well. So yeah, thanks so much, Shiny. I don't know if you had any follows up, or I can move on to the next person.
Yeah, thank you.
That's great, thanks. Maria is actually very interested in about if you have a paper or resource. Maria, do you,
Yes, thanks. Hi there.
or sorry, not Maria. I
Thanks
just
for
saw
your
that.
talk. I'm a biologist, just recently left Kaplan University, but I've been a part of the Valdez Islands Conservancy for a number of years, and we're very active doing a fair bit of research on the island in tandem with the Laxan Nation, whose land we're on. And we run a BioBlitz every year, and so we've been working on a intertidal survey on the west side of the island in Trincomalee Channel, which is extremely diverse, one of the most diverse areas that I have seen on the coast, and trying to pick up what's happened since the big heat dome of a few years ago. I saw Gloria's question about the sea star wasting disease. So there's so much that it's affected our intertidal shores. But I was curious when you talked about this approach to surveying that looked at these quadrats and identified them based on kind of the key species. I've never heard it addressed like that. It might just have been that you did that for the presentation, or is there something new that I've missed?
Yeah, I'll put the link in the chat. So
And was this also in relation to the drone work? Is that what I'm remembering? Yeah.
so the Marine group has developed protocols for monitoring biodiversity. Biodiversity methods and long-term monitoring methods for rocky intertidal habitats. And then we used a lot of the same methods as them and sort of adapted them slightly at our sites.
Okay, well, I'll have a look at that. Thank you.
Yeah, is that how, or did you have any like,
Well,
is
no,
that what
I'll
you
have a
wanted?
look
It's just,
and
yeah, I think
I'll
that
have a
would
look
be,
and see if it addresses my question.
I
I
think
think-
also I could be interpreting this wrong, but I think you're asking too about like focusing on a few key species with the quadrats. Is that one of the things you meant? Or no?
Could you just repeat the question? Sorry.
Oh, no, sorry. I was just trying to clarify what I understood of your question, which was in focusing in on a few key species from these quadrats. Is that what you were asking
Well,
about? Or
you're in the presentation, it was labeled as, anemone, barnacle, mussel, sea star. So,
yeah.
obviously we're looking at the zonation, and I can understand it as to why it's labeled like that. But I was just curious whether there was a different approach and you were actually not trying to capture as many species as we used to do.
Okay, yeah. Yeah, we did a bit of, I only shared that part, but we also did some biodiversity monitoring within the quadrat. So we did sort of a hybrid. When you go on the marine website that I sent, you'll see they have a biodiversity method as well. So we tried to we are combining their two methods so that we can look at changes in the distribution of those kind of indicator taxa. And then we can also look at changes in biodiversity. So yeah, I didn't really describe the biodiversity part of that, but it is important and a big part of what we do. Yeah.
And Michelle, are the names that you have here on the map, are they just kind of talking about the zone with which you're in, and then you do the biodiversity within those quadrants? Like you're not just looking at anemones in the green ones, or are you just looking?
Yeah, so I think we do the biodiversity, the reason I'm not certain is because so these were my quadrats. I was a sea star person. So last summer we set these up. So Sarah and Kaylee and a team of people were setting up these quadrats. So these will be permanent locations where we're always monitoring barnacles here, the abundance and taxa of barnacles, and then always mussels here and always anemones. And I'm pretty sure the biodiversity occurs in every one of these quadrats, not a subset. That's what I'm not absolutely certain about, but it does occur in each of these sort of specialized ones as well. And then these orange markers indicate the sea star transects, where I was digging through fucus and counting sea stars and looking for different like taxa and sizes, measuring the sizes as well.
All right, hopefully these resources are helpful. Mary.
Hi, I have a question for Mara about your Valdez surveys. I stumbled across the BioBlitz that the Galliano Conservancy bunch is doing, and your site in your link there is IMERS also. So do you guys use the same methodology?
So Andrew Simon took all the data that we had collected over several years, and with his pal in Europe actually put it on the website for us.
Yeah, that's why it looks the same as the Galliano one, because
Yeah, but
Andrew
we've
did that
worked
one
quite
too.
independently with that, and not only our own work, but I was given all the data from Andy Lamb and also the Vancouver Aquarium, and so it's a site that's been informed by quite a few different individuals.
And so are you collaborating with other groups? I mean, this ties into the work that you're doing, Michelle, with this long-term monitoring. It's ways of compiling all these different data atlases and spatial data sets that the NGOs have been doing. You know, there's lots of good work going on, and how does one find the compiled results from all these things?
Yeah, I think another thing I forgot to mention that was like of interest to us about this marine protocol is that you can compile the data and compare it to other projects that are ongoing in the US and in Canada. So Hackeye, they're using this marine protocol as well. And I think that in Canada, there may be others, but Hackeye is the only one that I'm aware of.
I think it was pioneered elsewhere on the coast, but that was some time ago. And I don't know what there was 3 or 4 sites that were established. To be worth looking into that. It was, 15 or more years ago.
I think one of the dilemmas when we're working with the marine environment is that the distribution of organisms is influenced by so many factors. And when somebody mentioned an indicator species, I know how easily that is applied to a land-based ecosystem. And I think of all the work we've done, and it's just interesting to see that we're still struggling to get all the networks connected, based on Mary's her comment there.
Yeah. Yeah, it can be a bit daunting, eh?
Yes, and I've been at it for years, and you're younger and doing some of the same things. So let's hope we just all have the energy to keep it going.
Yeah, part of what I love too is even with all the technology changing, like for example, the droning, so trying to get these big landscape scale studies, all that stuff we're still going back to the quadrat and ground truthing everything to the quadrat. We've all dug our
Yeah.
own quadrats. It's the best way to get the biodiversity, to get the counts, and it's just fascinating all this time later. So yeah, no, these are great discussions, and I just wanted to shout out both like from your projects and what we feed into and different things, but there is some online platforms I noticed that people are really trying to use, like the iNaturalist and that sort of stuff, to try and at least get data shareable and out there and people talking and communicating. Mary and Marja, sorry if I said your name earlier incorrectly. If you two are all right, I might go on to another question, but happy to keep going. Okay. So Michelle, not sure if you're going to be ready for this one. We'll give it a go. But sea star wasting disease. So can you make any comments
saw that
based on
one.
what you've seen for your kind of intertidal shallow coast research about the sea star wasting disease effects on the biodiversity? And if you have any specific examples, like are you seeing cascading effects for barnacles, mussels, seaweeds, or what you're seeing with sea stars? Or
Yeah, I don't think I'm really the person to comment on that one. But
okay.
just because like the only thing I have seen is that there are places where they're starting to come back, but this is just the beginning of this long-term monitoring project. So I don't have any like evidence-based comments or like information to share. I'm sorry.
That's
Do you
okay.
have
I think
anything, Heidi, or?
I am going to add a little bit just from the perspective that I used to do intertidal work, and I was doing intertidal work right around when the dying was happening, and it was significant. So for those of you that know, there was a sea star wasting disease that hit our coast, and just literally sea stars would catch this virus and start to like ooze and fall apart, and it was devastating for whole populations. Certain species did way worse than others. So for example, the sunflower star Pycnopodia is the hardest hit. It used to be, the range was from California to Alaska. It's technically extirpated, so like completely removed from its southern range. It is here still in BC and up into Alaska. But what they think exacerbated this, is like climate change, so warming waters. So what is fascinating is there's research coming out really shortly from some of our colleagues on the Central Coast. I think her name is Sarah Grabham. We'll look out for it. But what they found is that there were refuges in our deep fjords that had really cold water, and just that the steepness of the fjords that allowed them to either go deeper then or move up, that some of the Pycnopodia, these sunflower stars, actually avoided the disease, and they're starting to repopulate our coast. So that's a really exciting story that we're hearing about that one species on our coast. And then just a plug for the deep sea, because I moved to the deep sea, but 180 kilometers offshore of Haida, there's a seamount complex called the San Carlos Bowie Marine Protected Area. And 180 kilometers offshore, there's a seamount that reaches within 24 meters of the surface. So if you can get 180 kilometers offshore, you can literally go scuba diving on the top of a seamount. It's the craziest thing. And you can find coastal species there, like things that you would recognize. And we also have found that Pycnopodia, this other species, the species that did very poorly along the coast, the population is stable and fine out there. So very unique that BC has a couple refuge stories, as well as Alaska. And then I think the only thing that for intertidal people is, you're right, these sea stars are considered often what's called keystone species. So they're the large predators often, and they're shaping the whole ecosystem. So there definitely was effects and side effects from losing and these changing populations. But then, like Marcia said, there's so many things that affected. So not long after the sea star wasting disease hit, the heat wave and the blobs have hit. So blobs are actually weird terms that scientists have given to like warm water masses that just hang out and stay on our coast that haven't been happening more with climate change. And those have been affecting things like the intertidal habitat as well. So I think it's been really hard to study, but there are people out there doing it. Also, a site on Vancouver Island that appears to be a Pycnopodia nursery. That's really cool. I wonder what helped that refuge there. That's awesome. I'm just going to see, does anyone else have any other questions? I think, Mary, you actually asked at one point about the video that we were showing. Would that be classified as reef or not? So I don't know if Michelle would like to take over that.
Well, I just pulled up this image from one of Anya Dunham's CSAS papers about glass sponge. So, because it shows what the definition of a reef is when there is so much of that accumulation of sponge on top of previous generations of the sponge, which Heidi mentioned. And then the dead sponge gets like a silt settling in amongst it. And there's like layers and layers, and it builds up this structure called a bioherm, which is like, it says cemented by and buried in sediment. So it's underneath of the alive layer that's up on top. And these can be very large on the coast. But what we, and none have been, in the examples I was showing in Jervis Inlet, no reefs have been observed so far. And the way that they detect this subsurface bit is by using subsurface bioacoustics. So like this high frequency that gets because of this cemented part, the acoustics don't reflect as well off of it. So they get like a very different signal. And in the past, they've been able to use this subsurface tool to detect reefs down to about 20 meters across. So most of the large reefs on the coast have been documented using that technique. And then they're also ground truthed using visual surveys to see that this live sponge occurs on the top of them, or a damaged sponge. But in Jervis Inlet, what we saw was a combination of sponge growing on rock substrate. So that would, in higher aggregations, be called sponge garden. And we also saw these little pockets where we couldn't actually tell what the substrate was. So that could be because there's a small bioherm there, or it could be because there's just a lot of sediment on top of bedrock. So one of the recommendations that came out of this work is going to be that some subsurface acoustic surveys with higher spatial resolution than that 20 meters that I mentioned earlier are done in those areas to figure out what's going on, because there are some areas in Jervis Inlet where the available geological evidence, so like the older, larger, coarser spatial resolution, was indicating that they were possibly bioherm. And when we looked at them with the ROV, we saw a mix of dead sponge and live sponge, and we couldn't see any rock below. So those could be, we think, maybe dead reefs, but without that knowledge of whether or not they're reefs, it's difficult to know what management should do with them, because if they're dead reefs, they might have recovery potential. So the way that they treat them could be different. So yeah, I hope that answers the question of what a reef is. So a reef, just in summary, it's where you have that bioherm and where you have that evidence of like sponge growing on top of on top of older previous generations of sponge. And then another term that's often used is garden. So that's where sponge is growing on rock in higher kind of aggregations.
Great, thank you. Does anyone have any last questions before?
Sorry, it's
Mhm.
Shiny. I just have like 2 more questions, just 2 more quick questions.
Yeah, go ahead.
What questions about like why are the Olympia oyster why are they like important? Oh, sorry, not why are they like declining? And the second question is like the quadrant map you showed us. What's like the scale of it? Because I have no idea like how big it is. I was just wondering. Thank you.
Yeah. You're talking about the last map that I showed, eh?
Yeah, just
Probably of sponge and coral and rockfish. That's a 1 kilometer grid cell. And we didn't survey.
sorry,
that-
just the other map, like the one that's like where you have like the mussel quadrants and like the
Oh yeah.
yeah, that
Yeah, this is about probably 50 meters across.
okay.
Yeah.
Thank you. Or just
Yeah.
mm,
And your other question.
If you point out where the people or the gear is on there, that might make it more.
Oh yeah, totally good point. Yeah, there's backpacks, and these are little cones, the orange things. But yeah, backpacks are the best scale, I guess.
okay. Thank you.
Yeah, Shiny, sorry, what was your first question?
My first question is like why are Olympia oysters
Oh,
declining?
right. Yeah. Okay. Um, so I think it's one of those things where there's like several factors at play, kind of like the Pycnopodia. But the factors are so really heavy like harvesting before Pacific oysters, which are the ones that you see, the bigger ones that you see kind of everywhere on the coast now, before they were introduced.
Mhm.
Um, the Olympia oysters were, well, they were traditionally part of like indigenous people's diets, so they were like much more common um, and abundant. And then they became commercially harvested. You can look up in the DFO documents. You can, at one point I did that and saw that they were commercially fished quite heavily, and then their populations go down. And yeah, then like introduced Pacific oysters. I don't know what role climate change and uh, coastal development probably had, but they tend to um, the habitats that are important to them are like the estuarine habitats where people also like to occupy.
Yeah,
So
thank you.
if you want like a really good thorough description of Olympia oysters, there is it like in the Species at Risk Act, like the SARA registry, you can go in and like get all that background information.
Okay, thank you so much.
Yeah, probably.
Yeah, I think the biggest thing there was that we hit them hard and then it could put an introduced species in to outcompete them. And then they do have a little bit of niche partitioning, but then again, like what Michelle said, is that they're a little bit higher intertidal or near estuary. And so it's like this awful combination of then like habitat destruction and people moving, like, yeah, good old wham bam. Super fun.
Yeah.
Thank you so much for your talk tonight, Michelle. I got a lot of good conversation going, and I really liked seeing all the different tools. And I appreciate your time. And for everybody for being such a wonderful, engaged audience. I love it. I will just bring up that next month is another marine night on the last night of the month. So unfortunately, the calendar is not updated right now. So it's March 31st. It should be updated within the next couple days, but I'll just tell you it's another great talk. We've got someone here from the Adult Salmon Diet Program in the Wadis Lab at the University of Victoria, and it's a really cool talk about how they've been starting to use working with fishing people to use the contents of the stomachs of fish caught to like try and learn more about our coastal ecosystems. And it's just a really great talk, and yeah, so it will be a very interesting way to connect all the different pieces of biology. So another good one, come on out, tell all your friends. And yeah, if anyone has any questions, feel free to ask, but otherwise, good night. Have a great rest of your Monday and a good week.